Have you ever purchased a mini rose from the grocery store and then in 2 weeks it looks like this? Well, in this video I will show you how you can save your mini rose using plant tissue culture. So let's get started. The first step when taking your explants is sterilizing your tools. I use a glass bead sterilizer to heat my tools to 300 degrees Celsius and let them cool down before using them. In my case, I use alcohol to cool down my tools. As an alternative, you can also sterilize your tools using 20% bleach. You leave the tools in the bleach for a few minutes to kill any pathogens and rinse them to prevent the bleach from damaging the plant tissue. This plant is almost dead, so I'm taking as many explants as possible. However, I recommend not cutting more than 30% of your plants at once to allow it to recover. After cutting the main branches, I use my fingers to remove the leaves. This allows me to remove the leaves all the way from the base, which is not possible with scissors or a scalpel blade. Then I add my explants to multiple 50 milliliter tubes. They will all fit in one tube. However, I prefer not to place all my explants in one tube, as if one tube gets contaminated, I don't lose all my explants. Now it is time to sterilize the explants, and for that we need to make a fresh batch of 1 part bleach and 9 parts of water. Don't forget to add a few drops of surfactant so the bleach can do a better job cleaning the plant tissue. When I add my bleach solution, I don't fill the tubes all the way to the top. I like to give a gap so the plants can move around in the orbital shaker. Each plant is different and we require a different sterilization time, but I gave these mini roses around 20 minutes. Note that everything that you do from now on has to be done under aseptic conditions. So wear a face mask and gloves and spray everything with 70% isopropyl alcohol or ethanol. After sterilization, it is crucial to rinse the explants with sterile distilled water 3 times, each lasting around 5 minutes. Next, I take my explants using a sterile forceps and place them on a sterile paper plate so I can start cutting the ends off using a sterile blade. Trimming the ends of the tissue culture plants after sterilization is important for several reasons. The sterilization process, especially using a strong disinfectant solution like bleach, can damage the outermost tissues of the explants, and trimming them helps remove those potentially necrotic or non-viable parts of the plants, promoting healthier growth, and also the plants will uptake the nutrients and hormones much more easily if you trim the ends. Spray your tissue culture vessels with 70% isopropyl alcohol before placing the explants in the media. And don't forget to place the cuttings in the correct orientation. I know sometimes it's difficult to tell which side is up and which side is down. And if you place the plants upside down, it's not the end of the world. The plants will grow into the media, flip, and start growing up again. Introducing these dying mini roses to plant tissue culture will allow me to grow these plants in a perfect environment where they will grow happily. I don't need to worry about watering or fertilizing. However, this do not solve the mystery of why many plants you purchase get disease and die within a few weeks. Many plants sold in nurseries have pathogens already growing on them, and the main symptoms do not show up until they have already been purchased. Luckily, we can cure the plants from most pathogens using plant tissue culture and a technique called Apical Meristem, and this is how it's done. Using a stereo microscope, I cut the plant meristems, which means removing all the leaves from the plants. And I mean all the leaves. However, I need to be careful not to spread pathogens to the meristem, and to do that, you need to re-sterilize your blade after removing 1 or 2 leaves until you get to the plant meristem. Plant meristems are typically free from plant pathogens, especially viruses and some systematic bacteria and fungi. How? Meristematic tissues like shoot apical meristem has immature or absent vascular tissue, which means that systemic pathogens that move toward these systems cannot easily reach or colonize the meristems. Meristems have also multiple layers of protective leaf primordia or other tissues, making them harder to pathogens to reach the meristem. After removing the meristem from the plants, they also need to be placed in a sterile tissue culture media and wait until they grow back. This process can take several months, and the chances of success depends on the experience of the tissue culture technician. The plants will also need to be monitored and tested. Many viruses can be tested using a simple ELISA test similar to a COVID or pregnancy test. However, other viruses or viroids require analysis using a qPCR machine in a lab. These tests are regularly more expensive, but they can tell you much more about the plant's overall health. It's like going to the doctor, but for plants. So if you have a viable plant, I recommend testing it to ensure that it's free from pathogens. And this is how the plants look today. They have grown. They have been a couple of weeks and they have grown. Some of these leaves are not the most healthy because I believe these plants have some pathogens. So I had to wait several more weeks until the meristem that I took using the microscope grow back again, and the plants get big enough for me to test. So I want to run a test using a qPCR machine to make sure that the plants that are virus free, so I can start growing the plants once again. If you want to learn more about plant tissue culture and how you can cure your plants, subscribe to the channel. We have over 200 free videos to watch. However, if you want to learn more on how to do plant tissue culture in just 2 days, check out our tissue culture masterclass. The link is down in the description.